This study is conducted on the pre-limbic cortex for somatostatin neural function due to binge drinking in adolescents. The National Institute of Alcohol Abuse and Alcoholism defines binge drinking as repeatedly consuming alcohol, which increases the blood ethanol concentration. Somatostatin embodies a target for drug and alcohol use along with neuropsychiatric conditions. Somatostatin is around 20% of GABAergic neurons, which are cells in the basal forebrain in the rodent frontal cortex. Specifying their research, they focused on the prefrontal cortex since it is a vulnerable part of the brain for development and it is affected can produce lifelong damage. This study was conducted on rodents.
Before the experiment, the mice were weaned and moved into a temperature-humidity-controlled lactate circulation in the 4 week. After being in the dark for 3 hours, mice were fed 20% ethanol and tap water entering the dark cycle for 2 hours for 3 consecutive days. The 4th day, ethanol was presented for 4 hours, which represents the day of breakout. The straws were weighed at the beginning and end of each drinking window. Mice underwent 4 cycles of drinking in the dark, postnatal days, during which control mice had water on. The small mix of the first exposure day was to allow time lock electrophysiology and ligaments. Tail blood samples were collected 30 minutes after the last binge. Blood ethanol concentration was determined using an analog switch and a device to measure the enzyme oxygen uptake. The results show that the mice showed an increase in blood ethanol concentration above the threshold for binge drinking. Neuronal excitability was increased 24 hours after drinking. The results did not differ much in the sexes of the adolescents. The somatostatin excitability remained increased 30 days after drinking. Somatostatin neurons have been implicated in anxiety-like behavior studies assessing changes in anxiety-like behavior following adolescent alcohol have found mixed results, with some showing increases in anxiety-like behavior for adolescent drinking. With the results tested, there was enough evidence to provide increased validation for adolescent binge drinking as a clinically relevant model of binge ethanol consumption. We demonstrate that binge drinking during a broad definition of adolescence leads to increased excitability of PL